In the broad classification of microorganisms, the next type of organism is virus. First, we'll understand what are the general characteristics of viruses one by one. To start with, the definition. Viruses are defined as obligate intracellular parasites. We need to understand these meaning of words obligate, intracellular, and parasite. Obligate means it is a compulsory requirement of that organism. Intracellular means inside the cells, living cells, and parasite means dependency on other. So obligate intracellular parasites means these are the organisms who have a compulsory requirement of intracellular contents in order to reproduce. That means they can reproduce only within a living cell. If there is no living cell, viruses are not able to multiply or reproduce. That is, they do not have any metabolic machinery for reproduction. So they make use of the metabolic machinery of a living cell. And using this metabolic machinery, they can reproduce in large numbers in cell. Viruses can infect plant cells, animal cells, as well as bacterial cells. There are certain viruses which can also infect protozoa, fungi, and algae. Viruses lack metabolic machinery for their own to generate energy. So there is no as such energy generating mechanism in viruses. This means that viruses does not contain cytoplasmic inclusions or the cytoplasmic enzymes which are required for the growth and multiplication of viruses. They do not have machinery to synthesize proteins or cellular components on their own. Therefore, they are dependent on other cells. So, these cells are called as host cells. So, they are dependent on host cells to carry out their important functions. They do not replicate or multiply when present outside the cells. For example, viruses are present in droplets. Viruses may be present on dust particles. So, whenever they are present in this form, they are not multiplying. But if they get chance to enter into living cells, then they can reproduce within living cells. The structurally, they are structurally mature, infectious viral particles are called as virions. Viruses, in comparison to the size, are too small as compared to the size of a plant cell, animal cell, and bacterial cell. We measure the size of plant cell, animal cell, and bacterial cell, or fungal cell, or protozoa cell, or algal cell in micrometers, whereas the size of viruses is measured in terms of nanometers. So generally, virion size range from 20 to 300 nanometers. Therefore, they present the smallest and simplest of all infectious agents. There are certain definitions of viruses, for example, that they are acellular obligate intracellular parasite. So we have seen what is the meaning of obligate intracellular parasite. we need to add this word to this definition, acellular. Acellular obligate intracellular parasites. We have seen the structure of eukaryotic cell and structure of prokaryotic cell. So we have understood what component is called as cell. So a cell is having all the metabolic machinery and they can reproduce on their own. So what is this metabolic machinery? There are several cycles like glycolysis, TCA cycle, electron transport chain, hexose pentose pathway, and several other pathways which are going on, biochemical pathways which are going on inside a cell. So cell is a complex structure which contains cytoplasm and cellular inclusions. So structurally cells are complex as compared to viruses. But we cannot call virus as a cell because the metabolic machinery is absent in viruses. Therefore, they are considered as acellular organisms. At the same time, there is one definition of viruses, that is, virus is a link between living and non living. So, clearly, we cannot distinguish viruses as living or non living. The reason is simple, it shows few characteristics of living and few characteristics of non living. Whenever it is present outside the cell, it behaves as a non-living thing. But once it gets chance to enter into living cell, it reproduces. So therefore, it is also called as the link between living and non-living. This is the general structure of virus. So what we can see here is the most inside part is nucleic acid that makes the genome of that virus. So that nucleic acid, it may be either DNA or RNA. It can be neither. So both the types of RNAs, both the types of nucleic acids are not present in a single virus. It is either RNA or DNA. So this nucleic acid, it may be single stranded or double stranded. This nucleic acid is encased in a cover or capsule which is called as capsid. So this capsid and nucleic acid together, it forms a nucleocapsid. So this nucleocapsid which surrounds this capsid which surrounds this nucleic acid, it is made up with proteins, protein subunits. Capsids may have different structural symmetries, mainly icosahedral shape of capsid or helical shape of capsid. In this diagram, you can see icosahedral, a structurally organized, well organized structure called as icosahedral. And these capsid sometimes in some viruses may be encapsulated by one more envelope. So these viruses are called as enveloped viruses. Not all the viruses are enveloped. This is the general structure of virus. Capsids are made up with proteins called as protomers. These protomer units of protein come together and form the clusters of proteins. They are called as capsomers. In short, big units are capsomers and their subunits are protomers. These are the few different shapes of viruses. First one is the helical shape of capsid. All these are the shapes of capsid. It is the helical shape or rod shape. So here we can see the pink colored proteins that forms capsid in a helical form, and inside there is RNA or nucleic acid which is shown in red color. This is the typical structure of tobacco mosaic virus. Below that you can see the electron micrograph of TEM. This is how the virus looks like under electron microscope. They are too small, therefore we cannot make use of compound microscope. So we need to make use of higher resolving power having microscope, that is electron microscope. Second type of shape is polyhedral. So this is the organized shape. The green color balls are the proteinic subunits, and inside which the nucleic acid is embedded. This is the structure of adenovirus. So this is how it looks like under electron microscope. Another shape is spherical shape. You can see the spherical shape envelope, and inside that the shape of the capsid is helical. Pink colored uh, pink colored are the capsids, and inside which red colored ones are nucleic acids. This is the structure of influenza virus, which is closely related to coronavirus. Some of the viruses may have a complex symmetry like this. This is the structure of a bacteriophage, virus of bacterium. You can see the head is having a typical regular symmetry, which is called as icosahedral symmetry. And this is how it looks like under microscope. These are the different shapes of viruses which are formed due to the shape of capsid and the shape of envelope. This is what we are facing the problem nowadays. COVID-19 coronavirus, which is also called as SARS, severe acute respiratory syndrome, COV2, that is coronavirus. This is the structure of the coronavirus, spherical shape. You can see inside the pink color, it is RNA, and which is complexed with protein. So that pink colored threads are circular threads are nothing but nucleoproteins. So it is a helical shape capsid, and followed by that there is an envelope. So this envelope is very complex. You can see the yellow colored are spikes which are made up with glycoproteins. Blue ones are hemagglutinin esterase dimer, which are responsible for clotting of blood, which are responsible for bringing two red blood cells together. This is how this coronavirus looks like. Under electron microscope, coronavirus looks like this. Now, what is exactly icosahedral capsid? It is a very regular structure which is made up with proteins, and these proteins subunits are arranged. So how they are arranged? So it consists of 20 triangular facets. We can see in the picture what is the meaning of triangular facets. These are the triangular surfaces and 12 vertices. Vertices means the corners. Each triangular facet is an equilateral triangle. These facets come together to form 12 vertices. In icosahedral head, the nucleic acid molecule is highly compacted and folded. The right side figure you can see the electron micrograph. There are some viruses that possess either icosahedral or helical capsid, which is covered by envelope. These viruses are enveloped viruses. So there is nucleic acid which is surrounded by capsid, and this both are surrounded by one more envelope. Envelope viruses, the examples are mumps. This is the structure of mumps virus. We can see inside the blue colored nucleocapsid which is helical and which is surrounded by envelope consists of proteins, HN protein, F protein, M protein, etc. Measles virus. Measles virus, you can see inside the green colored wired nucleoprotein which contains nucleic acid and protein. So it is helical capsid, and this helical capsid is then surrounded by a complex envelope. This envelope consists of different proteins, fusion proteins, hemagglutinin, neuraminidase, HN protein, matrix protein, etc. You can see in this virus inside, which is attached to a nucleoprotein, is a polymerase. This is the typical feature of some viruses. It contains its own polymerase. Polymerase enzyme is responsible for replication of nucleic acid. So this organism, it uses its own polymerase for the replication of nucleic acid. This is the this is similar to the structure of coronavirus. So coronavirus also, it has its own RNA polymerase enzyme responsible for its replication. So all these envelope proteins are responsible for attachment and fusion with alveolar epithelium. Then rabies virus, it is a bullet shaped virus. The envelope is bullet shaped and the capsid, inside capsid is helical shape. It is also a RNA virus. So you can see inside yellow colored RNA, and it is complex with protein, nucleoprotein, and there is a surface in green color that forms the envelope formed by M protein, G protein, N protein, L protein, and P protein. These are the different shapes of viruses. One more example, herpes simplex virus. You can see inside icosahedral capsid, regular shape, and inside which double stranded DNA is embedded, and then it is surrounded by an envelope which is made up with glycoproteins. So this is again a typical structure containing double stranded DNA, herpes simplex virus, which is responsible for herpes disease called in Marathi it is called as Nagin. There are certain viruses which are called as complex viruses. Examples are Bacteriophages. Bacteriophages means viruses of bacteria. So the example here we are going to see is T even phage, which has an icosahedral capsid and tail fibers, base plate and contractile sheath. This is the beautiful structure of this T even phage. A capsid, you can see icosahedral capsid inside which there is double stranded DNA, the genome of this bacteriophage. Then there is collar sheath, contractile sheath. Why it is called as contractile? Because it can contract and relax just like an injection syringe. Then there is a base plate, and to that base plate the tail fibers are attached. This base plate also contains the spikes. So these spikes are responsible for spikes and tail fibers are responsible for attachment to bacterial cell wall. So these are the general characteristics. Viruses contain either DNA or RNA, but never both in the same. But other cells, eukaryotic cell and prokaryotic cell, they contain both DNA as well as RNA. The genome of a virion can consist of DNA or RNA, that is either double stranded or single stranded. Many of the viruses contain one or more enzymes like polymerase within their capsid that are released after the virus is uncoated in the host cell. The function of these enzymes is replication of nucleic acid of virus. The most common viral enzymes are polymerases. So the reproduction of viruses takes place by replication, in which the viral proteins and nucleic acid components are reproduced within the susceptible host cell.